Welcome to the Fankuchen Memorial Award. This year, David Goodsell is the awardee. The Fankuchen Award recognizes contributions to crystallographic research by a person known to be an effective teacher of crystallography. Isidore Fankuchen trained in Europe under W.L. Bragg and J.D. Bernal, and also collaborated with Dorothy Hodgkins on her work on steroids. After he returned to the US, he worked at MIT and then at the Anderson Institute for Biological Research in Minnesota. And he ended his career as a professor at the Polytechnic Institute of Brooklyn. His greatest influence was as a teacher and mentor to other crystallographers. Our awardee, David Goodsell, is a professor of computational biology, and this is a mouthful, in the Department of Integrative Structural and Computational Biology at the Scripps Research Institute. He's also a research professor at Rutgers University. David started working in the field of structural biology as a postdoc when he was working with Art Olson, where he developed an automated docking program that many of you may be familiar with called AutoDock. His most current research has involved developing a program called CellPack, which is a new method for creating 3D models of large portions of cells. For the last 2 decades, David has worked with the RCSB PDB, writing and illustrating the Molecule of the Month feature. And his unique illustration style is recognizable to all of us. His title of the talk today is Art as a Tool for Structural Biology, and his presentation is virtual, but I believe he will be on the line at the end to take audience questions.
Well, many thanks for that, that really nice introduction. You actually pretty much gave my whole talk there. So we'll go into that in more detail. So I'd like to start by thanking the nominators and the award committee for considering me for this award. I'm really deeply, deeply honored. And even more so because so many of my heroes are past recipients of this award. So I'm really, truly grateful. I'd also like to apologize right up front for not attending the conference in person. I was really looking forward to catching up with everybody. Unfortunately, I recently rediscovered that there are still viruses out there in the world other than SARS-CoV-2, and I really didn't want to share that with you. But I can share some of my work remotely, so we're good to go. So let me start the slides. Okay, so I'm going to talk a little bit for the next hour about some of the projects that I've done over the past 20, 30 years of using art as a tool for structural biology. So we'll start just with this little reminiscence or little look at what are the ways that we actually use scientific visualization, or in the more fashionable term these days, sci-art. And so I'm sure everybody in the room there uses visualization pretty much every day here at work. We all use computer graphics to look at the structures that we're solving and figure out what they're doing and everything. So we're constantly using computers and visualization in our research. During this talk, I'll also talk about some projects that I've worked on where I use art to integrate a bunch of different types of data into one coherent view, which is maybe something that, that you haven't done yourself quite as much. So visualization is also super important for science communication. It's the way that we talk to each other with our images in journals and movies and animations in these types of presentations. It's also super essential for education and outreach, just to bring the structures that we're solving to life for people that might not be experts in structural biology. Finally, at the end of this talk, I'm going to talk about a few examples where I've done visualization just because it's loads of fun to do. Okay, so I'll start with a little bit of ancient history here, just talking about how I started using visualization and art as a tool for the science that I was doing. And so way back when I was a grad student, over here, this little glamour shot up in the corner shows me with my graduate advisor, Dick Dickerson. And I was trained in crystallography with him, solving structures of little tiny pieces of DNA. And at the time, we had this brand new, the department had this brand new multi-picture system, interactive, one of the first interactive computer graphic systems that had just become available, and affordable by a whole department. But unfortunately, nobody knew how to use it, which of course was an advantage for me. So I sat down to start coding up some ways to use that for the structures that we were solving. And so one of the easiest things, of course, was down here in the corner to come up with these dot surfaces to show van der Waals contacts. In our system, this is the DNA base. You can see over here, and here in the minor groove is a trial anti-cancer drug that packs really tightly in the minor groove. So that was a nice way to display that. I also, as everybody else did at the time, used ORTEP for just about everything. And this is down here at the bottom is a corrupted version of ORTEP that I added all of these extra lines to show the contacts between helices and the crystal lattice. Over here is a raster image. Raster stuff was brand new at the time. This image actually took about an hour to calculate on our VAX computers. And then finally, for systems that were beyond my coding ability at the time, like these packing diagrams at the top, I just drew those up by hand for journal figures. So I was never particularly good at growing crystals. So I then focused on the computational and graphic stuff basically for the rest of my career, and started that by moving to Scripps Research to work with Art Olson in his molecular graphics laboratory, which was just an amazingly creative environment for developing new methods to use the technology of the day to look at molecules. And up here in the corner is a picture of Art with some of his augmented reality stuff he's been doing lately. And down here is just an example of one of the things I did as a postdoc, which was to look at advanced methods of doing volume rendering. So this is a picture down here using these kind of glowing cloudy volumes to look at a difference map of one of these anti-cancer drugs in the minor groove of DNA. And basically all of the other stuff that I did with Art in that postdoc led to everything I'm going to talk about for the rest of the talk. So just kind of looking at today, we all enjoy a really wonderfully user-friendly toolbox of methods that we can use basically every day that we're studying molecules. And I'm sure we're all very familiar with all these different options that are available out of the box with most of these turnkey molecular graphics tools. We use wireframes when we want to look at chemistry, and we look at backbones when we want to simplify things, and CPK when we want to look at how things fit together and what the holes look like, and ribbon diagrams to look at protein folding, and basically just for the wow factor. I mean, they're so incredibly beautiful. But this toolbox might not be as useful when we go to start talking to people who are not experts in structural biology. And that kind of can be exemplified by going to the Nobel Prize site, to this page called Information for the Public, which I find it remarkable that I usually do this as a guessing game at talks. Most people are not going to be able to recognize these as molecules or understand what the diagrams are trying to say the way that we as structural biologists understand what each of these representations is showing. To me, really the only picture that a person out on the street is going to understand is this picture of DNA over here. The rest of it is just a bunch of wiggles. So back when I was a postdoc, I tried to help come up with solutions to this problem of creating interpretable images of molecules that might be more generally understandable by people outside of the field. And so I was at a SIGGRAPH conference, a big computer graphics conference, and saw this paper presented, which came up with ways of creating very cartoony, accessible images from computational models. And so I ran home and coded it up. And so it uses this, at least part of the algorithm uses a very simple method. What you do is create a Z-buffer map here. So things that are dark are further away, things that are white are closer to us in this image. And then you just draw a line wherever there's a big difference. And so it creates a picture that is just like something I would draw if I was going to do this in pen and ink, that pulls out all the big features of the molecule, throws away all the little tiny details and intersections between every atom. Like all postdocs, I ran back home and kind of went crazy for the next couple of months and coded up all kinds of crazy stuff, all these contour lines and highlights and who knows what. But it all kind of settled down to the style that I've been using ever since here. And so this style is built from these very accessible, easy, cartoony outlines that give a real familiar look, and then nice flat colors to show the different subunits between in these larger assemblies. And so, using this coloring scheme and these simple outlines, it's easy to see this clothespin, right, sticking on the DNA and the enhanceosome, and easy to see the different subunits of this adenovirus without having all of the individual atoms get in the way of that understanding. The atoms are still there, so you can still show somebody that an atom is this big compared to the whole protein. But the individual atoms are not the major story that you're trying to tell with the picture. And I'll just put up this picture down here with the little question mark. Do that as a guessing game to yourself. This is going to be the molecule of the month in September. And I can give you a little hint that the coloring scheme is set up so that the brighter colors down here are things that are encoded in the mitochondrial genome. And the lighter stuff is from the nuclear genome. So preview of coming attractions. Okay, and so for many, many years, this program basically had a user community of one. I used it for all of my outreach stuff. And recently, a talented developer in the lab, Ludovic Ouden, put together a nice little web interface for this. So now it's, so you can orient your molecule using this little NGL window and then press a few buttons and get an image out. It's also nice to see that the style has been adopted by a number of programs now. So there are instructions out there for doing in Chimera, and Molstar has some hardwired hooks to create similar illustrative images. So, yeah, go. If this is something they want to play with, there are a lot of opportunities to use it. Okay, so moving on, I'm going to talk about two large-scale projects that I've been doing basically for my whole career for the past 3 decades. One of them is this idea of creating images that integrate experimental information to try to create a depiction of the cellular mesoscale of molecules inside living cells. The other big goal has been to tell these structure function stories about individual molecules as part of the outreach effort at the RCSB Protein Data Bank in the Molecule of the Month column. So I'll talk a little bit about that first and then go on to the cell pictures. So the Molecule of the Month is intended to be a really friendly introduction to structural biology, to ease people in, to use dynamic, colorful illustrations to get people interested, and then also include links into the main PDB archive, hopefully to entice people to go in and start exploring molecules in more detail themselves. And so I've been doing this for like 22 years now. So there are lots of different examples in here in the archive of articles that I've done. But I'm always amazed that there are still really basic things that are yet to be done. There are so many great molecular stories to be told. And now with the revolution of in cryo-EM, these stories are getting bigger and bigger and bigger. So it's going to be really fun to continue for the next 20 years. So I do a few things in these to try to make them a little more appealing to novice users. The first and most important thing to me is to reduce the jargon in these columns. And so that can be visual jargon with the way that I described earlier, these simple, colorful illustrations that are more about the individual proteins and how they fit together, and written jargon as well. So I try to keep the text very short so people could get in and out quickly with lots of links to other resources in the PDB 101, and also right back into the main PDB archive to look at the actual structures themselves. Another thing that we try to do is to choose topics that can provide, that have a lot of personal context so that people can understand why learning about the structures of these proteins and molecules are important for health, for general science, for biotechnology, stuff like that. And so to exemplify that, here are 6 topics that we chose last year for a boot camp that we did with undergraduates and graduate students, where we brought the students in and they formed teams and each wrote an article and illustrated it. And so we chose a diverse collection of things here. Some things related to health over here, nanopore sequencing related to biotechnology and design proteins, biotechnology, protein design. Everybody wants to know about hemoglobin, right? As a central thing in teaching of structural biology, and big, cool basic science structures like this ribonuclease P that's involved in the maturation of tRNAs. Okay, and the final thing I always try to do is to, if we can, somehow show structure-function relationships, processes, something to show how we can get multiple views of molecules and build up a story for how the molecule works. And so, I mean, this is a very obvious one taken from a recent article centered on cancer, the Warburg effect and pyruvate kinase, just showing the allosteric transitions in this enzyme. And so we always, at the end of the article, put this section called Exploring the Structure, where we bring in a few structures from the archive and kind of create a worked example of how you might use really traditional molecular graphics to explore some aspect of that protein's function. And so here I've just provided 3 buttons in JMOL here that let people flip between the 3 allosteric states that we're providing in the JMOL. And hopefully that'll give people hints about how they can start exploring structures themselves. Okay, I also, related to processes, I'm trying to do more and more pictures like this that tell larger stories instead of stories about just one individual molecule. And so this is a picture I put together as part of a series on exploring the structural biology of cancer, just showing a whole signal transduction network. And the fun part of this one is that all the little globular bits, of course, each one has a PDB structure, and I'm using AlphaFold here for all the wiggly bits in between. To me, that's one of the big advantages of AlphaFold, is that it makes you look at the whole protein structure, you know, even the parts that aren't necessarily ordered or well-determined. Okay. And so this molecule of the month column lives in a big ecosystem of educational materials at PDB 101 at the RCSP Protein Data Bank, which is put together by a really wonderful team, a team of outreach people led by Christine Zardecki. And so this includes flyers on topical things like the SARS-CoV-2 genome here, activities like this fun little game that was done as part of the 50th anniversary of the Protein Data Bank, educational animations by Maria Voigt, full curriculum modules by Shuchi Dutta, that people can just slot right into their classrooms. And then topical things like recently we've been doing a lot of pandemic outreach and education, as you might expect. So I invite you, if you haven't already been there, to visit this PDB 101 site and take a look at all the goodies that we have to offer. Okay, so we'll move on and talk a little bit about the paintings. And so I started this work a number of years ago with the hypothesis: is it possible to create a structural model of a living cell or a piece of a living cell that includes all macromolecules and integrates the experimental data that's available at the time? So the goal is to create an illustration that has all the molecules, the right size, the right shape, interacting in the right way, in the right place, the right number of them, all of that stuff. And so when I started doing this back in the early 90s, the answer to that question is almost. So I started with the system that really had the most information at the time, which was E. coli. And this is what integrative structural biology looked like back in 91, okay? This is what proteomics looked like, okay? It's a big 2D gel. You squish up the cell, run it out on this gel, and then go through laboriously one spot at a time and try to figure out what the protein is. And of course, you can use the intensities more or less to give you information about the abundance. This is what PubMed looked like at the time. So of course, I needed to do a lot of legwork, dig up a lot of different information from the primary literature. But this Science Citation Index was the only way to go forward in time from one article to see which articles had cited it. So it was a really interesting treasure hunt of trying to dig down papers, going backwards from references in the papers and going forward with the citation index. The PDB at the time had a couple hundred structures, right? So the ribosome was still in the future. A lot of really central things were still in the future. The ATP synthase, all of those fun things were still yet to be solved. So things like ribosomes, I had to draw based on cryo-EM, which at the time gave a very blobby general contour for these molecules. So these are the small ribosomal subunit from James Lake. Really wonderful stuff. So over the years, I've used E. coli as my kind of central test system for this hypothesis and updated it year after year after year based on the current state of knowledge and my own ability to find that information. So, I can only include information that I can find. And as the speaker spoke this in the morning session, I don't have an infinite amount of time. And so here's that image from 1991 in Tibbs. A few years later, I did this picture for American Scientist, moving to color, which shows the different functions and compartments of the cell more clearly. Down here, 2009, this was for my book. At this point, every molecule in this picture has a name. In previous ones, for a lot of the soluble things, I just had to use distributions of sizes and oligomerization states. But by this time, I was able to actually come up with a name for every molecule in the structure. You can see the flagellar motor is still pretty much made up. And down here, just last year, I did an updated illustration. And at this point, there's a pretty good structure for just about everything in this painting in the PDB, either structures from E. coli or based on close homologs. So it's getting better and better and better. So for instance, you can look back in this old one. This is what how I drew HU protein, which is protein involved in the DNA packaging. So I mean, drew it kind of like a nucleosome because we didn't know too much better. And in more current ones, it's based on the crystal structure of this little scissor-shaped molecule that bends the DNA. Okay. So this is what the process of integrative structural biology, at least in the context of me putting these paintings together, looks like today. Tons and tons and tons of information, right? So I was able to find, this is last year, I was able to find a full proteome for E. coli with abundances. So I was able to start at the top of that list, put in those abundant proteins, and then dot in all the other cool ones in between when I still had space. The PDB has just about everything that you'd ever want to see. Uniprot helps you make sense of all of those PDB structures in terms of sequence, in terms of function, in terms of where they sit in membranes, all the information you need to make sure that everything's in the right place. And PubMed, the world's literature is at your fingertips. So this process of chasing down information is much, much, much easier. And so just over here, one of the total wonders of the PDB, there are so many, is that this flagellar motor now, we're just this close to having a full atomic structure of the whole thing. It's absolutely mind-boggling. Just spend just 2 minutes here talking about some of the artistic decisions that I have to make in these illustrations, these paintings, cell paintings, to help make them a little more interpretable, because they're super, super complex, right? So I made these choices early on as a way to make them clear. So perhaps most importantly, I do everything in orthographic projection. So these aren't perspective views, where you're immersed in the middle of the cell with molecules all around you, which are perfect, for animations and immersive views. But I chose early on to use this orthographic projection so that I could show a big chunk of the cell, and so that people can compare molecules and the size of these ribosomes to the size of enzymes to the width of the DNA. I want to make all those things at the same scale and comparable. I do a lot of artistic license in the choice of molecules. So I put in all the abundant ones at their concentrations, like the ribosomes, and the amount of DNA, and then dot in all the ones that I want to highlight in between those, the ones that are less prevalent, less abundant. I also use a lot of artistic license in the choice, in the way I do the cross-section of this. So for instance, the membranes are always normal to the clipping plane so that you can see the shape of that membrane. It's never clipped obliquely. And the DNA is all done in the plane of the page. I never have DNA helices coming towards you and being clipped, because that makes for a very complicated picture. And so it's very much an approximation. This isn't a super accurate model of how that DNA is arranged in the cell. No small molecules. The picture would be just even more of a mess if they were included. Very simple shapes and flat colors. And I like to be able to have people make a comparison between, for instance, this enzyme, also fructokinase, the atomic structure of it, and this little butterfly in the illustration. And so using a similar outline flat color style, I hope makes that transition a little easier. I include subunits sometimes when they're telling an interesting story, like in this chaperone, or all the subunits of the dehydrogenase complexes. But sometimes they just get too complicated. So for instance, with ribosomes, I typically just do the large subunit and the small subunit. Depth skewed very sharply to black to help with depth relationships. And then the colors these days are chosen to help highlight the functional compartments of the cell, so people can separate the cell wall from the soluble cytoplasm area from the nucleoid area. I did a different coloring scheme early on, where I colored all the proteins one color, all the lipids one color, all the nucleic acids a different color, and the pictures were just a mess. So I went to this compartment-based one quite quickly, and it seems to be working a bit better. Okay, so somebody always asks a question about the process of doing one of these paintings. So I'll just spend a couple slides here talking about the process of painting it. This is a picture I did right at the beginning of the pandemic of coronavirus. At the time, there wasn't a whole lot of information about SARS-CoV-2. So this is a picture that's more based on the previous SARS. And when I write captions for it, I write coronavirus as opposed to being SARS-CoV-2. And, it's interesting. We see so many pictures of SARS-CoV-2 these days. I'm sure everybody out there can look at this picture and say how this picture is wrong if we were to call this SARS-CoV-2, like things like the number of spikes and how rigid they are and all of that. So the most time-consuming part of doing these illustrations, of course, is digging up the information. But fortunately, it's tons of fun. It's always like a big treasure hunt to try to find enough information to support all the different aspects of the virus or the cell that I need to depict. And so for this coronavirus picture, there was a beautiful structure for the spike, although missing the transmembrane portion. I had been doing some modeling at the time of the nucleoprotein complex in the middle, some really nice cryo-EM data to give the size of the virion and distribution of spikes. And then, unfortunately, for some things like the membrane protein, all you can do is go to Uniprot and make sure that it's the right size and has the width of the transmembrane segment is consistent with the number of helices that are crossing. So there's always going to be some artistic license in these illustrations. And here's the process of creating the painting. I do a full sketch here in pencil to build in all the foreground molecules. That's transferred to watercolor paper using carbon paper, which carbon paper is tricky to find these days. I'm using some that my grandfather gave me many, many, many years ago. And then I just start painting in each molecule one at a time using a little tiny brush. Here, the membrane is in. If you squint, you can just barely see it. And the spikes are in this brighter magenta. This one down here has most of the foreground done, and then finally filling in the background and putting in some ink outlines to clean the whole thing up. This kind of underscores an interesting part about the style that I use, which is that in this early one here, you can see that membrane is super light, right? And so in this image, you can barely see it. It's not the major part of the illustration. But after all of the background is put in and the depth cueing, it ends up being one of the most prominent parts of the illustration. And these pictures are really fun to do because those kind of tonal relationships aren't apparent until I do the very last little piece of black in here. So they kind of all of a sudden pop and emerge. It's a really, really fun process. Okay, so I have two big goals with these paintings. The first thing and foremost thing is really that I want to inspire some of the wonder that I feel about structural biology in people that are seeing these. And so, this illustration of E. coli is just filled with amazing stories. The one that came as a surprise to me, I knew nothing about until I started researching it, is this little protein up here at the top, which acts like an elevator to carry the lipopolysaccharides from the inner membrane where they're synthesized to the outer membrane where they have a function of protecting the surface. You can't make this stuff up. It's so incredibly amazing. And I've also been trying more and more to incorporate molecules that are intrinsically disordered in these paintings, since it's becoming more and more apparent that that's absolutely an essential part of all of protein function inside cells. And so that includes the replosome here, which is held together, all the bits and pieces with these wiggly chains, and these little arms that come off of the ribosome and help organize the various factors that are needed. This degradosome here with a bunch of enzymes all held together with floppy bits. And it just goes on and on. Just many, many interesting molecular stories to tell while putting these kind of paintings together. The second thing I've been trying to do more recently is to use these paintings as a challenge to the modeling community, and saying, a lot of people are, including ourselves, are saying we're at the stage now where we can create models, 3D models, accurate 3D models of entire cells. And so I'm throwing these illustrations up as a challenge and saying, well, this is the kind of stuff that you're going to need to model if you want to make these models accurate. And the reason I think these paintings are an effective way to pose this challenge is that, for me, painting these things, it's really rather easy to prototype new types of interactions between molecules. I can draw anything I want in these things. And so as an example here, this is a painting I did of a casein micelle, which is really my first attempt at trying to paint one of these liquid phase separation systems that are becoming apparent that they're so important in cells. And so I was able to look at all of the conflicting models for what this micelle might look like, and then try to come up with a conception that fits the size of the molecules and how people think they're connected together, the level of flexibility that's going to be needed if you wanted to build a full model of these things, and all of that. So there it is, a challenge, posing a challenge to the community. And so in my lab and Art's lab, we're picking up this challenge now and building some of our first models, 3D models of entire cells or organelles. And so we've started with a very well-defined system, mycoplasma cells, which have been modeled using a systems biology approach with Marcus Covert. And so they've modeled the whole cell, the replication, the translation, transcription, number of molecules, enzyme reactions, everything. And so basically they can just hand us a recipe of how many molecules need to be included and what's bound to the DNA, everything at particular time points along the life cycle. And so we can use that as a recipe and then say, here's a structural snapshot of what that time point looks like. So this is work that was done by Martina Martin during her postdoc, and a very talented programmer or a developer in the lab, Ludovic Outen, who's been putting together all the tools for building these kind of models. And so these are the kind of pieces that we're bringing together to build these models. Martina spent 2 years putting together a structural proteome for the whole cell, combining a very small number of experimentally determined structures with homology models when that was effective, and homologs for things like ribosomes, where homology modeling really isn't possible. And so this is what her final proteome looks like. She has it colored here by a confidence score that she developed, and perhaps not surprisingly, the membrane proteins, she's much less confident in those than she is in the soluble proteins. So I dusted off my coding skills to come up with a coarse grain model of the nucleoid. And so this uses a lattice-based method that's fast enough to make it possible for a supercoiled DNA, RNA being transcribed, free RNA, spheres to say where we need to put polymerases and ribosomes and stuff like that. And finally, Ludo put together just a beautiful set of pipeline for creating these models built around this online tool that he's calling Mesoscope, which is a platform for bringing together all of these different structures, curating them, making sure that they're structures that you want, defining the coarse grain representations that you need during the packing, and all of the little tasks you need for integrating these information before you build a model. And then he's built the tools, CellPack GPU for creating a coarse model, relaxation in NVIDIA Flex, or recently he's been relaxing them in lamps, and finally exporting it so that you can look at it in molecular graphics programs such as MolStar. So here's a little movie that Martina put together. If you saw Art's talk this morning, this is all going to look very familiar. If this is very choppy on your screen, I invite you to visit our website, and there's a nice gallery of images and animations that Martina put together to show off her several years of work in constructing this model. And these are the kind of scientific games that we're playing right now with these kind of models. So in the Covert Lab, they looked at a bunch of different properties of these cells over the course of the lifetime, like how long did it take for a particular gene to be transcribed the first time, or what's the abundance of a particular protein expression. And so we're able to go into these models and then color based on all these different properties that that Covert and his colleagues are interested in looking at. Okay, and related to this, recently I've been extending this work to create some models of an even simpler cell, the Venter Institute Syn3A cell, minimal cell, in collaboration with John Glass and Zan Uthi-Shulman and Elizabeth Villa, electron microscopist. And so, as I often do, start with a painting just to define all of the different things that we're going to need. This is actually the first painting I've ever done that includes an entire proteome. So this includes every protein that's made by this made by this cell. Here's a draft of a 3D model based on that, and I also had the great fun in this this project of there were 100 structures that were annotated as and uncharacterized in the proteome, and so I went to AlphaFold and spent a really enjoyable couple of weeks predicting structures for each of those. And you know, some of them came out. This looks pretty good to me, right? You know, glycolipid synthase looks enzymy here, and membrane bound looking. This spectrony thing, really cool. You know, maybe it's true. Bound to the membrane here with a transmembrane helix. A lot of the things came up like this, just little bits and pieces that formed one helix. Who knows? Maybe these are subunits of a larger complex. And then we got for a few of the larger ones, got the typical spaghetti when it didn't really know what to do with it. I'll just go really super quickly through this because I want to talk about a little bit of the fun stuff at the end. We're also working on models of a whole organelle. This is from pancreatic beta cells, picking the characteristic organelle in those cells, which is this insulin secretory vesicle. It has one of the few examples of a functional protein crystal that has a function inside of a cell, you know, for storage of the insulin. The work was done by Brett Barbero and Ludo. A proteome. The proteome was interesting for this one because there were 3 experimental proteomes available, which had a grand total of 6 proteins in common. So we had to develop a protocol for deciding which of the other proteins we thought were actually in these vesicles, and we used annotations from Uniprot to help sort that out. And then these are the kind of games that we're playing with these models we're getting X-ray tomograms from Carolyn Larabelle, who's part of this pancreatic beta cell consortium, looking for bright blobs in that, which are presumed to be the secretory vesicles. So this is a segmentation of all those blobs, and then creating a whole panel of different sizes of vesicles and levels of maturity, all the way from immature to totally mature vesicles and transitional forms, and then matching up computed X-ray tomogram images with the experimental tomogram images to try to classify which of these vesicles are mature and which are immature and what their sizes are, stuff like that. So this, I'm hoping this is kind of where the cell organelle modeling is going to go as a tool to help interpret experimental microscopy data. Okay, Art also mentioned this program, Cell Paint, that we put together a couple years ago. The goal of this program is to help put these kind of tools into people's hands that maybe don't have time to spend several weeks creating a painting by hand. So it takes its lead from a traditional digital painting program, where over here we have a panel of molecules, and then the user can just paint in membranes and pop in membrane proteins. The membrane proteins know that they're membrane proteins, so they go into membranes, move stuff around. Here the membrane protein of SARS-CoV-2 is going in, painting in some plasma molecules out here, and then finally at the end, increasing the temperature and letting everything just bump around a bit and create a more interesting picture. So we, with Cell Paint, we did a contest a while back at the RCSP Protein Data Bank, gave people two goals. One was to create educational stuff about SARS-CoV-2 and got really nice entries back like this up in the corner, and then also just play and have fun, right? So here's a picture of Rosalind Franklin all done in molecules. Where's the other one? Somewhere in here, down here is a Pac-Man all made of antibodies chomping down on a virion, you know. So this is just testament of, if you give people the tools, there's so much creativity out there. You never know what you're going to get back. And then here's just our first attempt at trying to use tools like this as a way to help interactively interpret electron micrographs. And so here's mitochondria, and just dotting in a few molecules based on features that you can see in the micrograph. Work in progress. Okay, so I'll just finish up with a few little fun projects that I've done over the years to stay sane. So I had the opportunity a couple of years ago to do a summer residency at the JRC Resident Artist Program. Wonderful thing. They bring a handful of artists and scientists in together, and you get a month to do nothing but create. Really amazing. And so I focused on origin of life and put these two big large-scale paintings together. Over here is a conception of the RNA world and self-replicating RNAs. And this one over here is the last universal common ancestor, so the cell that gave rise to all life on Earth, at least my conception of what it would look like, basically with the hypothesis that the big development that this cell had over all the other ones is that it had a consistent way to segregate its DNA between the two daughter cells when it's dividing, with these cool little motors that squirt the DNA in two different directions. Some fun stuff over here. You can probably guess this painting was titled Cytoskeleton, which I did for a t-shirt a little while back, and had the opportunity to learn some stuff that I didn't know a lot about, like up here at the top, the multi-synthase complex that brings together a bunch of tRNA, amino acyl tRNA synthetases into one big complex. And who knew, these big expansion segments of RNA on the ribosomes. So our ribosomes have these big floppy arms all sticking out in all different directions. Who knows what they do? Interesting stuff. Over here is a steampunky conception I did of protein synthesis for a project that was talking about cells as computational Turing machines. And so this was a lot of fun. I managed to sneak my signature up here, DSG, and put Turing's name here. Unfortunately, there's not a U, of course, but V is just as good. There's a project I did with a PDB to celebrate the 50th anniversary of the Protein Data Bank. I also did this, I have to admit, during the election season a couple years ago, and it was all so discouraging that I needed something to distract myself and get excited about. And so this is the thing that came out of that. The face cards over here follow people through a career in science. So starting with maybe a high school intern up here and ending up down here with a gray-haired professor, much like me. I've also been doing an ongoing series trying to create just a tiny pushback to the mountains, the flood of misinformation on vaccines. And so I've been doing a series of paintings on vaccines. Over here is a influenza subunit vaccine binding to a B-cell surface, B-cell receptors here on the surface. And over here is a simplified conception of the SARS-CoV-2 mRNA vaccine. I've also been updating paintings as information has become more and more available on SARS-CoV-2. So here there's that original coronavirus one I did at the very beginning of the pandemic. This one is a conception of the fusion event releasing the nucleoprotein here from endosomes into the cytoplasm, and starting with the translation down here. And the most recent one here, a journal cover showing neutralizing antibodies bound to the surface of SARS-CoV-2 with our nice bendy spike proteins, and hopefully a better conception of what the nucleoprotein looks like, although everything I've seen so far is still pretty blurry about that. And so I'm also very, very lucky to, through my association with the RCSP Protein Bank, Data Bank, to be able to make all of these paintings freely available, free for use for whatever thing you'd want to use them for. So I invite you to visit the gallery over there. All these paintings are available, large files, and free for any kind of use you'd like. So based on that coronavirus picture, we did a little coloring exercise right at the beginning of the pandemic when we were all locked down and everybody was scared. Nobody knew what was going on. So we created this coloring page, dropped it out through PDB 101, and got some really wonderful responses back on Twitter. In particular, there were a bunch of people that came back and said they were using this coloring exercise to sit down with their kids during the lockdown and have a nice friendly exercise activity where they could talk about the virus and hopefully help make it a little more real, like it's a real object, exists and can be fought, and so on. Okay, finally, in the last 2 minutes, I'll just flash up a few pictures I've been doing for fun, trying to get back to my roots as a crystallographer. And I've been exploring the PDB again and pulling up structures and drawing pictures of crystal lattices, because who doesn't like crystal lattices and crystal symmetry and all of that? So here's the one that started it all, the crystal lattice of myoglobin. Here's an amazing tetragonal portal program from a portal protein from a phage. Cannonballs stacked up with ferritin. These are found treasures, right? I just go to the PDB and try to find interesting packings. And this absolutely amazing nucleosome structure with these snaky DNAs all wrapping around and around and around and around. Found treasures, like I say, it's just super fun to explore the PDB. Okay, so I'll finish up just with a few acknowledgements. I want to send a shout out to Art, who gave a really beautiful talk this morning, and thank him for his super creative environment at the Molecular Graphics Lab, now expanded to be called the Center of Computational Structural Biology with the 4 PIs. And also send my deep gratitude to Stephen Burley, the director of the RCSP Protein Data Bank, Helen Berman, the director emeritus, and a special shout out to Christine Zardecki for overseeing all of the amazing things that are happening at the in the outreach section of PDB 101. And over here support from NIH for the cell modeling work and support from NSF, NIH, and DOE for the RCSP Protein Data Bank, continuing, allowing it to still be a free resource that's available to anybody. And I'll just put up this announcement. The PDB is looking for people for a number of positions. And I can tell you personally that the RCSP Protein Data Bank is a wonderful, wonderful place to work. Really a vibrant environment full of very creative, talented people. So I invite you to look at that. Many thanks. If there's time, I can take a question or two.
So Andy Howard, Illinois Tech. Something like 1 of 14 American males are red-green colorblind. Do you find some productive ways of responding to that in terms of maximizing information intake for people like me?
Yes, for sure. And Andy, hello to you. I think we crossed paths way back when I was at UCLA. So yes, there are a bunch of methods for previewing how images will look for colorblind individuals. And I don't use those as much as I should, but I definitely try to avoid any case where there's red next to green for something that I want people to see. I can say personally that there are a bunch of color palettes out there that are specifically designed for people with various levels of colorblindness, and they're just okay at best. And so I actually would love to have the opportunity to explore that in more detail and try to come up with palettes that would be aesthetically pleasing to you and to me and to everyone. So my answer is yes, I try, but I do what I can do.
Thank you very much.